The biosynthetic pathways that produce amino acids and the bases of nucleotides are long and complex, requiring a large investment of energy by the organism. The cell therefore needs a signal to tell it to stop producing more of a particular compound. The signal is frequently part of a feedback inhibition mechanism, which we will examine in this lesson. Recall that in feedback inhibition, the end product of a pathway inhibits an earlier step in the pathway, most commonly the first step. Although this type of energy conservation makes sense in any case, it is particularly so in the long and complex steps of the biosynthesis of amino acids and nucleotides. Glutamine synthetase is a key enzyme in amino acid biosynthesis, and its activity is observed in all known organisms. As you can see from the reaction scheme below, it generates glutamine by the transfer of an ammonia group to the side chain of glutamate. Note that this reaction costs energy in the form of one molecule of ATP, and that the enzyme requires magnesium ion as a cofactor, as we've observed for almost all nucleotide binding enzymes. Glutamine is the primary amine carrier in most organisms. Occupying such a key role, it is not surprising that this enzyme is highly regulated in its activity. As you can see from the schematic below, glutamine can be converted to the amino acids serine, histidine, and tryptophan, as well as other compounds. It is therefore feedback inhibited by these compounds. In addition to being feedback inhibited by these varied compounds, it is also inhibited by glycine and alanine, key markers in amino acid metabolism, including the amino acid serine. We will find that glutamine is also the starting point for generating other nitrogenous compounds, whose buildup will also inhibit this enzyme. Hopefully from this brief overview you can see how key is this enzyme and how glutamine is truly the primary amine carrier in cells.